Okay, thank you, Rudy. So I'd like to welcome all of you again to this open session of the National Advisory Council for Human Genome Research, including those of you here in the room and those joining us remotely. As with the rest of the open session, my director's report is being video recorded, and that recording will be made available as a permanent archive on NHGRI's website, genome.gov. For those who might be new to our council meetings, I want to make you aware of the electronic resource that has been developed for my director's report, analogous to a supplemental materials associated with a published paper, and the resource can be accessed at the URL that's shown on this slide. And noted and circled in red is the fact that you can also access these slides that I'll be showing both in PDF and PowerPoint formats. We have created this electronic resource with an electronic dimension such that when there is a relevant document or a relevant website we wanted you to know about, we have created this website that is numbered by document numbers, and the slide would be indicated with a document number in the bottom right. All of this stuff, the video, all of these documents, the whole website will be permanently archived on genome.gov as a historic record of this council meeting. Now, there will be a number of other presentations during the open session of this council meeting. My director's report will try to steer away from going into any detail about what other speakers might cover. Following my presentation, you will hear from NHGRI's scientific director, the person that oversees our intramural research program, Charles Rotimi. He's going to give an update about our intramural research program. That has deliberately been timed because after lunch, Council members Gal Jarvik and Lynn Jordy will present the NHGRI Intramural Research Program Blue Ribbon Panel Report, and I'll tell you more about that just before Charles's presentation. This will be followed by a presentation about the recently released NANSEM report on the use of race, ethnicity, and ancestry as population descriptors in genetics and genomics research, and we're fortunate to have the co-chairs of the group that developed this report will be here in person. Aravinda Chakravarti and Charmaine Royale. So that will be the first set of presentations, but that's not all, because we will also have a presentation that will serve as an update on the H3 Africa program that will be given by Jen Troyer, one of our extramural program directors. And lastly, we do have work to get done here that council needs to help us with and vote on. We have two concept clearances that will be given by extramural program directors. First, Rob Rowley is going to give a concept clearance on genomic-enabled learning health systems. And Xander Arguella will present a concept clearance on developing novel theory and methods for understanding the genetic architecture of complex traits. So that's what we have in store for us during the open session. And now I'm going to go through these 7 areas in order that really nicely give me an opportunity to give updates about what I think are the main areas that are worth covering. And I'm going to start with some general NHGRI updates. So NHGRI just released, and I mean literally just released, the Building a Diverse Genomics Workforce Action Agenda Implementation and Progress Report. Now, the purpose of this report is to articulate NHGRI's progress on our recent programmatic efforts to diversify the genomics workforce. The plan details the programs, projects, initiatives, and engagements that have been enacted since the release of our action agenda in 2021, and the strategies articulated include a number of partners and continued engagement with colleges, universities, professional societies, and industry. And diversifying the genomics workforce cannot be achieved without continued support from the scientific community. And so we really invite you to read this progress report, which is available on our website, genome.gov. And if you have any thoughts or input about it, we certainly want to hear from you. Now, the Human Genetics Scholars Initiative is a program developed with the American Society of Human Genetics, or ASHG. It also involves NHGRI and other partners that aims to help advance diversity and inclusion in genetics and genomics. Now, this program identifies and mentors a select group of diverse early career individuals, helping them achieve professional success. And in April, more than 20 predoctoral, postdoctoral, and early career human genetics scholars visited NHGRI in person During their visit, they had the opportunity to interact with me, extramural program directors, and intramural researchers to discuss research funding and career development opportunities. For example, I gave an update about NHGRI's new initiatives and upcoming events, and I also discussed future directions for the Institute. Now, NHGRI has supported this initiative since 2019, and we plan to make this an annual in-person event with these scholars and future scholars. Now, among many things that got delayed by the COVID-19 pandemic has been the ability to get group photographs. And in fact, I will warn you that we are overdue in getting an updated council photograph, so that might be on the to-do list for the September meeting. Maybe we'll get it with the cake, and we'll make it a real celebratory thing. Well, in any case, I am immensely proud of my leadership team at NHGRI, which is called the Directors Working Group. And this distinguished ensemble meets on a roughly weekly basis and helps me in every possible way to lead NHGRI with virtually all important decisions made with their involvement. So not surprisingly, the composition of the Directors Working Group changed a bit during the pandemic, but getting every member of the group in the same room at the same time took the better part of a year. But we finally did it, and we were able to capture this wonderful group photo, which I wanted to share with you. And I would just say, besides being incredibly distinguished looking, these people are individually and collectively amazing leaders who I am deeply indebted to for their commitments to genomics, for their dedication to NHGRI, and for their friendship. So with that, I will move on to some general NIH and NIH-related updates. And I guess I'll start with a related update because Rochelle Walensky, who is actually going to now step down as director of the Centers for Disease Control and Prevention, or CDC, on June 30th. I think all you recognize the CDC as one of our sister agencies within the Department of Health and Human Services. Rochelle took the helm of the CDC in January of 2021 and helped to guide President Joe Biden's response to the COVID-19 pandemic, really from his first day in office. During her tenure as director, she launched a reorganization of the CDC to modernize the agency and improve its reputation. She also declared racism a serious public health threat and led the agency in its efforts to contain a multinational monkeypox outbreak. Maureen Goodenow has transitioned from her former role as the NIH Associate Director for AIDS Research and Director of the NIH Office for AIDS Research to a new role that she has as a Senior Advisor in the NIH Office of the Director. Maureen will continue to serve as the Chief of the Molecular HIV and Host Interactions section within NIAID's Intramural Research Program. She was the first woman to serve in this position since its establishment in 1988. Bill Campagianis will serve as the acting NIH Associate Director of AIDS Research and acting Director of the NIH Office of AIDS Research while a search for a new director is conducted. In another transition, Karina Walters, an enrolled member of the Choctaw Nation of Oklahoma, has been selected as the director of the NIH Tribal Health Research Office. She succeeds David Wilson, who is now on assignment to the White House Council on Native American Affairs. Karina is a social epidemiologist and a health prevention scholar with more than 28 years of American Indian, Alaska Native health research experience. She has conducted social epidemiological research on the environmental, historical, social, and cultural determinants of health and health equity in American Indian and Alaska Native communities. Earlier this month, Carrie Wallenitz departed from her role as Senior Advisor in the NIH Office of the Director. Carrie joined NIH in February 2015 as the NIH Associate Director for Science Policy, where she oversaw broad policy issues related to clinical trials, human participant protections, biosecurity and biosafety, and data sharing and management policies. In April of 2021, Carrie accepted a detail to the White House Office of Science and Technology Policy, or OSTP, where she served as the Deputy Director for Health and Life Sciences. And she is now at, or will soon be at, a Washington, D.C.-based government relations firm. In March, NIH released its NIH-wide strategic plan for diversity, equity, inclusion, and accessibility, or DEIA. The 5-year DEIA strategic plan covers fiscal years 2023 to 2027 and articulates NIH's mission to integrate DEIA in all activities in pursuit of the agency's mission. The plan specifically prioritizes NIH's role as a people-centered organization and encourages the NIH community to enhance DEIA in its operations, workforce, and research. The plan includes cross-cutting themes for promoting transparency, communication, and engagement, fostering sustainable change, and harnessing data. The plan applies to the internal and external NIH workforce and can serve as a roadmap for NIH-supported institutions as they advance their DEIA efforts. Now, meanwhile, NIH is seeking input on their proposed changes to the National Research Service Award, or NRSA, fellowship review. There have been some persistent concerns that NRSA fellowship reviews disadvantage some highly qualified but promising applicants. As a result, a Center for Scientific Review, or CSR, Advisory Council Working Group was convened and recommended that both the review criteria and the fellowship applications be revised. The goal of seeking additional input about these initial recommendations is to further improve the review of NRSA fellowships and to identify highly promising scientific scientists of the future, all while ensuring fairness and equity for all applications. Those interested in submitting feedback must do so through a formal request for information, or otherwise known as an RFI, by June 23rd of this year. In March, President Biden released his federal request outlining the administration's proposal for federal spending in fiscal year 2024, which begins on October 1st. The president's budget is an indicator of the administration's priorities, and its release marks the opening of the budget negotiation season. Now, most NIH institutes and centers, including NHGRI, did not receive an increase in the president's budget. Rather, NHGRI is proposed to be flat compared to the current fiscal year, with the additional funds proposed for NIH going mostly to programs like ARPA-H. It remains to be seen, though, what the House and the Senate will propose and agree upon. So, as always with budget increases, I leave you with stay tuned, and we will see what develops. In terms of some general genomics updates, we have a number of sad things to report. Paul Berg, a Stanford University emeritus professor of biochemistry, died in February. Paul shared with Wally Gilbert and Fred Sanger the 1980 Nobel Prize in Chemistry for his fundamental studies of the biochemistry of nucleic acids with particular regard to recombinant DNA. He created the first recombinant DNA molecule, which marked the dawn of the genetic engineering and the molecular biology revolution. Casey Brown, associate professor of genetics at the University of Pennsylvania Perelman School of Medicine, passed away in March. He was a brilliant scientist whose innovative work centered on approaches to understanding how human genomic variation influences gene expression. He was a pivotal member of the Genotype Tissue Expression, or GTEx, consortium, and was a gifted, deeply informed, and highly charismatic teacher who was beloved by students and trainees. Janet Sitzheimer, who also passed away, it turns out, in March. And Janet had a long and prominent career at UCLA, where she was a professor of human genetics, biomathematics, and biostatistics. Her research focused on developing mathematical models of evolution and genetics. However, her passion remained teaching, and she was very involved in NHGRI's T32 program and served as a doctoral advisor for 13 students. Charles Scriver, Alva Professor Emeritus of Human Genetics at McGill University, passed away in April. He was a pioneer in the field of medical genetics, with his research focusing on a subset of rare genetic disorders called inborn errors of metabolism, and his efforts spearheaded Quebec's innovative newborn screening program for congenital diseases. He was a tireless teacher, and his work improved the lives of countless children and their families around the world. Matt Hurls, a longtime NHGRI collaborator, has been appointed the new Wellcome Sanger Institute director. Now, Matt has served as the head of Human Genetics program at the Sanger Institute since 2017. As many of you know, Matt, his work focuses on deciphering the genetic causes of severe developmental disorders and understanding how DNA mutates through the generations. He was elected a fellow of the UK Academy of Medical Sciences in 2017 and a fellow of the Royal Society in 2019. The National Academy of Sciences recently announced their newly elected members, and of particular relevance to NHGRI and the genomics community are the people listed here, including people at NIH and people outside of NIH, and we would just want to extend our congratulations to these friends and colleagues. And then in March, the National Academies of Sciences, Engineering, and Medicine, or NASM, released the report Using Population Descriptors in Genetics and Genomics Research, a New Framework for an Evolving Field. And this report is a product of a study conducted by an international committee of expert volunteers that NASM assembled and charged with reviewing and assessing the existing methodologies, benefits, and challenges in using race, ethnicity, ancestry, and other population descriptors in genetics and genomics research. NHGRI was one of the 14 institute centers and offices who were co-sponsors of this study, and NHGRI served as a task co-leader along with the All of Us research program for this study. The report details past and current uses of population descriptors and proposes guiding principles and a new framework for the field. And I'm not going to tell you more about it because later today, Aravinda Chakravarti and Charmoine Royal, the study's co-chairs, will be here to discuss the report's findings and recommendations. That wasn't the only thing NASM was doing. In another NASM effort, a distinguished committee has been convened to conduct a 2-year consensus study to determine what scientific and technological breakthroughs, workforce, and infrastructure are needed to directly sequence RNA and its modifications. Now, the study is sponsored by the Warren Alpert Foundation, the National Institute of Environmental Health Sciences, and NHGRI. And the committee actually hosted a public workshop in March as part of their information gathering process. And the next major opportunity for input from the community will be through an ideation challenge that will be held June 12th through 14th, where interdisciplinary teams will participate in structured brainstorming and problem-solving activities in order to identify and solve the key challenges in this important scientific area. Moving on then to our extramural research program, and it will start with a real bang from last week. It was exhilarating but exhausting at the same time. The Human Genome Reference Program, or HGRP, represents NHGRI's continued commitment to advancing and maintaining the Human Genome Reference Sequence. Now, a draft of the highly anticipated Human Pangenome Reference Sequence was released this month. Actually, it was released last week. The Human Pangenome Reference captures significantly more population diversity than previous references. Currently includes genome sequences from 47 people. So with each person carrying paired chromosomes, the Human Pangenome Reference currently contains 94 distinct sequences. And researchers in the Human Pangenome Reference Consortium are aiming to generate 700 reference sequences by 2024. Now, the Human Pangenome paper is one of several recently published in Nature by consortium researchers. The companion papers report the study of the evolution of acrocentric chromosome short arms and segmental duplications, the analysis of inversion polymorphisms, and a tool for constructing and analyzing pangenome graphs. The Nature cover, which I don't know if you've all received yet or have seen, which is shown on the slide on the right, depicts the pangenome wrapping around the globe and uses a sequence tube map rendering of a pangenome relating 10 haplotypes within the highly variable HLA-A locus on human chromosome 6. And the map was created by Adam Novick at the University of California, Santa Cruz, and the graphic itself was generated by NHGRI's Daryl Leisha. Now, the news coverage, this is the exhausting part, the news coverage about the new Human Pangenome Reference began in earnest on May 10th, one day after a press event involving nearly 80 journalists. And this is one of the most highly attended briefings that the journal Nature has ever hosted. The coverage was reflected by more than 600,000 news mentions and had a total reach of more than 1.2 billion, according to online circulation numbers of the word pangenome. Now, top outlets, and just to give you a tease of the kind of things we saw, was major coverage by the New York Times, by the Washington Post, by CNN, by the BBC, by NBC News, by Reuters, and on and on and on, and we could have showed you many more. And all of this covered the announcement, which really nicely helped spread our message of the importance of diversity in genomics. And I even got to participate in a few live radio and TV interviews, including this one for Sky News and another one for Al Jazeera that came out over the weekend. So it was a great amount of very positive attention to the field of genomics last week because of these publications and the news coverage that it garnered. Now, one effort stemming from NHGRI's investigator-initiated portfolio is the Zoonomia project. Now, this project, which aims to catalog the differences across mammalian genomes, recently published 11 papers in a special issue of the journal Science with a way cool cover on it. The Zoonomia project is an international collaboration involving over 150 researchers that is using comparative genomics to study the genomes of over 240 mammalian species. NHGRI has been a major funder of the Zoonomia project, contributing about $2.8 million over 4 years in an R01 award that has supported genome sequencing and analysis. And in this special issue of Zoonomia researchers, a special issue of Science, Zoonomia researchers report a number of findings, including specific regions of the mammalian genome that have remained largely unchanged through millions of years of evolution. In addition, NHGRI K99R00 awardee Stephen Riley and his group reported how loss of some of these conserved regions may play an important role in uniquely human traits. Other researchers summarized how Zoonomia has helped to understand the biological links across species, which can lead to the development of diagnostics and treatments for humans. And finally, Beth Shapiro and colleagues used comparative genomics to provide insights into the selective pressures that shaped Balto. And Balto is a heroic member of the sled dog teams that helped deliver life-saving diphtheria medicine to Nome, Alaska in the 1920s. Moving on, the Centers of Excellence in Genomic Science, or CEGS program, supports interdisciplinary research teams working together to develop highly innovative approaches in genomics research. Now, since my last update about this program, one center was successfully renewed, the Center for Synthetic Regulatory Genomics, led by Jeff Buca. The center will expand their technologies for generating synthetic DNA molecules that are 50,000 to 100,000 base pairs long, which is being termed big DNA. They will also develop methods for studying those big DNA molecules in stem cells and humanized mouse models in order to improve our understanding of gene regulation, genomic variation, and human disease. Now, the CEGS funding opportunity has been reissued with the next receipt date on June 23rd, 2023, followed by a receipt date of June 21st of 2024. Now, in terms of one of our newer programs, last year, NHGRI launched the Molecular Phenotypes of Null Alleles in Cells, or MORPHIC Consortium, which aims to develop an extensive catalog of the molecular and cellular phenotypes for null alleles for every human gene. Now, in its first phase, the program will optimize available methods to knock out 1,000 genes, evaluate the utility of various cell lines and organoids, explore multiple molecular and cellular assays, develop analyses to understand the use of and requirements for the data, and finally to disseminate the results to the research community. The MORPHIC Consortium is made up of 4 data production research and development centers at the Jackson Laboratory, Memorial Sloan Kettering, Northwestern University, and the University of California, San Francisco. There's also 1 data resource and administrative coordinating center at the University of Miami. And applications are being discussed actually during this May Council meeting for the final component of the program, the Data Analysis and Validation Center, which will undertake data analysis and modeling to evaluate and help ensure the use of MORPHIC data. The consortium's first in-person meeting took place, by the way, earlier this year, back in January and February. In the fall of 2021, NHGRI, together with the Eunice Kennedy Shriver National Institute of Child Health and Human Development, the National Institute of Neurological Disorders and Stroke, the National Institute of Mental Health, and the Office of Research Infrastructure Programs, launched the Development of Genotype Tissue Expression Consortium, a mouthful, which is why we call it DGTEX, which aims to establish a resource database and associated tissue bank to study gene expression patterns in multiple reference tissues during human and non-human primate development. Now, NHGRI, along with our partners, recently published a new funding opportunity for data integration and statistical analysis methods. Applicants are expected to propose the development of innovative and generalizable statistical and computational methods for DGTEX data analyses. There is also strong encouragement to develop methods that assess the influence of genomic variation on development, compare gene expression and regulation across tissues and time points, or leverage comparative genomics to understand developmental and evolutionary processes and integrate with existing tissue and cell atlas efforts. And applications for this new funding opportunity are due on November 20th. The NHGRI Genomic Data Science Analysis, Visualization, and Informatics Lab Space, another mouthful, which we simply call ANVIL, is a federated, cloud-based infrastructure and software platform that provides an analysis and a computing environment for genomics research. ANVIL distributes genomic and phenotypic data through both unrestricted and controlled access. These data sets include over 600,000 genome sequences and more than 5 petabytes of total data, most of which are from NHGRI-funded programs and initiatives. The ANVIL group has been working to expand outreach efforts to broaden the awareness and also to help educate new users about ANVIL. One such effort was a presentation at the Advances of Genome Biology and Technology, or AGBT meeting in February, which focused on making genomic data science accessible to researchers from diverse backgrounds through ANVIL. Now, genomics relies on resources that make data, materials, and tools widely available and accessible. Now, these resources are essential for researchers to facilitate their understanding of the genome and to explore its role in health and disease. So, recognizing this, NHGRI continues to invest in genomic community resources by renewing its funding opportunity announcements, which will support the development and distribution of resources that are valuable for both basic and clinical genomics research. Examples of resources include collections of analysis tools and software platforms, community data standards and ontologies, collections of samples or biological materials, and comprehensive genomic data generation. The first round of applications for the renewed funding opportunity is due on May 25th, and the next round after that will be in September. Now, NIH has also released notices of intent to publish 2 trans-NIH funding opportunity announcements to support data resources. The first will support early-stage data repositories and knowledge bases, while the second will support established data repositories and knowledge bases. And the earliest estimated application due date for both of these is sometime in the fall. The NHGRI EBI Genome-Wide Association Studies, or GWAS catalog, is an online resource providing curated results from GWAS papers and user-provided summary statistics. On April 28th, the GWAS catalog celebrated its 15th anniversary. The catalog started in 2008 with a single study on age-related macular degeneration. Now, 15 years later, the GWAS catalog contains over 515,000 curated associations from over 6,300 publications. There have been over 60,000 user-submitted summary statistics, as well as over 5,800 citations of the GWAS catalog and associated papers written by catalog investigators. The number of studies and associations has increased over the years, suggesting that the initial investment in GWAS continues to yield dividends, even as more advanced genome sequencing technologies become widespread. Also shown here on the right is a cake that was baked in honor of the iconic GWAS catalog diagram, which nicely reflects, although not to any realism, the density of data that's there. If you tried to do that in a cake, it would get a little too sweet probably or something. Well, in any case, we wish the GWAS catalog a very happy anniversary. In July of 2020, the Electronic Medical Records and Genomics, or eMERGE network, comprising 10 clinical sites and a coordinating center, launched a nationwide study to evaluate the impact of genome-informed risk assessments, or JIRAs, on participant and clinician behaviors and decision-making. Now, to create GERA, the network validated polygenic risk scores in diverse populations and combined the scores with other forms of risk, monogenic risk, family health history, and clinical covariates. And in doing all of that, and then return the result of the risk of developing 11 common complex diseases. Now, the eMERGE network has begun returning results to patients and providers at 5 of those sites, returning 753 results to date. Of those, 156 are high-risk results, which are provided with specific recommendations for clinical action. And after this, the network will assess the uptake of these recommendations by patients and providers. The eMERGE network also published a marker paper in January of this year entitled Returning Integrated Genomic Risk and Clinical Recommendations, the eMERGE study that came out in the journal Genetics in Medicine. The paper describes the design of the ongoing eMERGE study that I just told you a little bit about, and the network aims to return roughly 1,500 results this year. The Clinical Sequencing Evidence Generation Research, or CSER program, aims to generate evidence supporting the clinical utility of genome sequencing with a major emphasis on utility in diverse patient populations and healthcare settings. As the CSER program concludes its work this spring, investigators have been busy presenting and publishing their work. So at the 2023 American College of Medical Genetics and Genomics, or ACMG annual meeting, there were 10 oral presentations and 9 posters given by CSER researchers. The topics included fetal exome sequencing, clinical genome sequencing tools, and ethical considerations of reproductive genetics. This slide highlights actually 3 of the oral presentations given by CSER investigators. First, there was Teresa Sparks, who reported on differences in genes implicated in disease, comparing prenatal to pediatric patients. Hadley Smith reported on the development and validation of a new CSER-wide outcome measure to assess the impact of genome sequencing on children and adults. And finally, Mary Norton reported findings on prenatal clinical exome sequencing in a study where 64% of the participants were from an underrepresented population. CSER investigators were also busy publishing other papers, in this case, a couple of papers that were related to lessons learned by the consortium. The first paper examined the challenges that CSER investigators faced during the COVID-19 pandemic. The authors interviewed representatives at each CSER research site and identified factors that affected each site's experiences with the COVID-19 pandemic. The second paper focused on identifying pragmatic challenges related to designing and implementing research that involves underrepresented populations. Moving on to a different clinical program, our Clinical Genome Resource, or ClinGen, evaluates and disseminates information about the clinical relevance of genes and genomic variants for use in precision medicine and research. And as a global resource with members from 59 countries at present, ClinGen aims to provide training on its resources to encourage worldwide participation in ClinGen curation efforts. So, for example, an ongoing partnership with the Human Heredity and Health in Africa, H3 Africa program's Rare Diseases Working Group has yielded 3 workshops to develop accessible educational content for African investigators. The most recent workshop, held in March, focused on variant classification using ClinGen's variant curation interface. And previous workshops were held in 2021 and 2022. And shaded in blue on this map of the African continent are the 23 African countries that have participated in at least one of these workshops, with 7 of these countries actually participating in all 3. And in total, the workshops have attracted over 450 participants, some of whom have become active members of the ClinGen expert panels and working groups. Now, meanwhile, the image shown on the right shows ClinGen PI and former council member Sharon Plawn leading a training session during the annual conference of the Society for Indian Academy of Medical Genetics in December of 2022. During this hands-on session, participants learned about ClinGen variant curation guidelines and how to perform variant classifications. The Polygenic Risk Methods in Diverse Populations, or PRIMED Consortium, is developing methods and refining polygenic risk scores, or PRS, to improve the prediction of disease risk in diverse ancestry populations. Primed has at least 32 publications in print or in press, and displayed here on the right is a figure from a Primed paper that highlights the goals and impact that Primed efforts will have in optimizing polygenic risk scores for assessing coronary heart disease risk in diverse populations. And the figure illustrates the steps in the PRS development process, specifically A, generating large and accessible data resources by assembling and sharing GWAS data from diverse cohorts. B, developing novel methods for ancestry-optimized PRS and creating public resources for curation. And then going on to C, creating reference PRS frameworks using population-based cohorts and estimating reference-adjusted scores. And finally, D, integrating with clinical algorithms to incorporate genetic and clinical risk factors and calibrate the risk models for clinical translation. And the Primed Consortium's Methods Review Working Group has also transitioned to a newly formed Methods Development Working Group, which will lead state-of-the-art PRS development approaches in diverse populations. And finally, the Primed Consortium and the eMERGE Network appropriately held a joint meeting recently to identify areas of mutual interest and collaboration for the advancement of polygenic risk score development and clinical implementation. The Global Genomic Medicine Consortium, which we call G2MC, was established by NHGRI and the National Academies of Sciences, Engineering, and Medicine back in 2014. And this group is going to hold their 7th international conference in October of 2023 in Geneva, Switzerland. It will actually be a hybrid meeting convened on Campus Biotech, which is known as the Swiss Center of Excellence in Biotechnology and Life Sciences Research. The title of this year's conference is Genomic Medicine for Everyone, Accelerating Implementation of Genomic Medicine for Global Equity and Access. There will be 3 themes for the conference: implementation of genomic medicine, collaboration, and diversity, equity, and inclusion, or DEI, in genomics. These themes are based on the WHO Science Council's report calling for accelerating access to genomics for global health. Registration for the conference is actually now open for those interested. NHGRI's Advancing Genomic Medicine Research, or AGMR, funding opportunity is now in its 3rd year, encouraging applications that stimulate innovative and advanced understanding of when, where, and how best to implement the use of genomic information and technologies in clinical care. And that these are also broadly applicable to genomic medicine and are significant beyond a single disease. Now, the research topics funded to date include implementing genomic medicine for undiagnosed and rare disease patients, integrating clinical decision support and other tools into electronic health record, or EHR systems, building the evidence base for clinical and economic value of genomic testing, screening of newborns for genetic disease, and promoting access for and understanding barriers to genomic medicine in underserved populations. Now, a notice of intent to publish, or NOIP, has been released for the renewed funding opportunity, which is expected to be published this summer, and will have an accompanying notice of special interest, sometimes referred to as a NOSI, for soliciting applications from small businesses through the parent R43, R44. The Technology Development Coordinating Center, or TDCC, facilitates opportunities for grantee collaborations, leads efforts to promote standards in genomic technologies, develops resources to share with the research community, and finally disseminates information about program advances. And the TDCC has organized a virtual outreach series sharing insights from industry leaders and genomic technology developers and users. And the objective of this outreach series is to provide post-secondary students, postdoctoral fellows, and biomedical researchers with a fundamental understanding of how these new genomic technologies are utilized, as well as to showcase the opportunities and challenges associated with their optimization and their use. The sessions will be held over the next 2 weeks and will focus on emerging technologies and methods, such as direct RNA sequencing, base modification analyses, single molecule protein sequencing, advances in single-cell genomic analyses, and methods for studying genome-wide regulatory mechanisms. Now, each of these virtual events is free and open to the public. However, you do need to register, and so registration is required. NHGRI's Small Business Program continues to support commercialization and technology transfer by U.S. companies working across the genomics landscape. A unique funding opportunity is available for early career academic scientists through NHGRI's Small Business Program. Now, the Small Business Transition Grant is designed for individuals interested in transitioning to entrepreneurship. The mechanism also supports the transfer of technology from academic laboratories to small businesses. This program uses a fast-track mechanism where participants start with a Small Business Technology Transfer, or STTR, award in Phase 1 before transitioning to a Small Business Innovation Research, or SBIR, award in Phase 2. Mentorship is required, and entrepreneurship training is provided. The program was released in partnership with the 3 other institutes that are listed here, and applications for this are now due on August 21st. The Training and Career Development Program aims to prepare a talented and diverse genomics workforce by providing both institutional and individual funding through a variety of mechanisms, including individual fellowships and career development awards, institutional awards, and diversity supplements. Programs are offered at the undergraduate, post-baccalaureate, graduate, and postdoctoral and fellowship levels. And shown here and summarized is information about the 8th Annual Training and Career Development Meeting, which was hosted by Duke University, but it was actually held in Salt Lake City in April. And I'm sure Lynn Jordy, our council member, immediately recognizes that Skyview as his place of origin, where he actively works. But the meeting brought together, in the beautiful backdrop of Salt Lake City, roughly 300 in-person attendees, and also included 50 virtual attendees. The Training and Career Development Program would also like to highlight 2 notices of funding opportunities, or NOFOs as they're called, focused on enhancing workforce diversity. The entry-level module, or ELM, for training the genomics research workforce support the development of modules of genomics-related curricula for those in the entry-level research workforce. Entry-level, by the way, in this case, is defined as positions that do not require a bachelor's degree or higher. Applications are due June 1st. The NIH Science Education Partnership, or SEPA program, supports informal science education activities designed for the pre-kindergarten to grade 12 levels. These activities aim to enhance the diversity of the biomedical, behavioral, and clinical research workforce, as well as to foster a better understanding of NIH-funded research and its public health implications. And applications for a SEPA award are due July 14th. And here are some notable highlights from NHGRI's extramural investigator-initiated research portfolio. First, Daijiang Liu and colleagues developed a tool called TESLA, which integrates GWAS summary statistics, whole genome sequencing data, and multi-ancestry EQTL data. They applied TESLA to tobacco use phenotypes and identified 273 new gene associations. They also performed in silico analyses and found several drugs that may be repurposed to treat nicotine addiction. Second, Paul Kaveri and colleagues developed a framework integrating gene expression, chromatin accessibility, transcription factor footprinting, and DNA looping data to identify over 800 cell-type-specific recurrent DNA motif combinations, also called DMCs. They found that neoplastic cancer cells were activating DMC processes that healthy cells did not. Finally, Kate Spector-Baghdadi, an investigator funded by an NHGRI Mentored Research Scientist Development Award, and colleagues surveyed genetic researchers on the use of shared data resources and identified key factors that facilitate the efficient utilization of these resources. These factors include accessibility, harmonization, and administrative support. Moving on to the NIH Common Fund and other trans-NIH activities, starting with the Undiagnosed Diseases Network, or UDN, which, as many of you know, is an NIH Common Fund program that aims to improve the level of diagnoses for rare and undiagnosed conditions through a team-based approach. And over the past decade, the UDN has received over 6,300 applications and accepted over 2,500 participants and provided clinical evaluations, genome sequencing, model organism genomic variant screening, and metabolic analyses. The network has successfully assigned diagnoses to over 670 participants, and that number continues to grow. Since its inception, the UDN has made great strides in discovering novel disease genes, gene variants, and syndromes, and in connecting these to disease pathways. From this work, the UDN has published over 200 manuscripts, submitted over 600 genomic variant interpretations to ClinVar, and shared just under 2,000 records in MatchMaker Exchange. But with 10 years of NIH Commons support ending in June of this year, that's next month, the UDN has transitioned to being a trans-NIH program led by the National Institutes of Neurological Disorders and Stroke and supported by the 16 institutes and centers listed here. The vision for the next phase of the UDN is to evolve into a larger self-sustained network to provide expert diagnostic services for undiagnosed patients and to foster scientific discovery. The Data Management Coordinating Center was awarded to Harvard Medical School to provide infrastructure, data management, and clinical research support for the next 5 years. And clinical sites will join the network as diagnostic centers of excellence, allowing them to access the resource, giving them access to resources to assist patients with undiagnosed diseases. Now, a newer NIH Common Fund program is the Bridge to Artificial Intelligence, or Bridge to AI program, and that is really aiming to set the stage for the widespread use of artificial intelligence, or AI, in healthcare research. Now, the goals of this program are to generate flagship AI-ready data sets centered around biomedical grand challenge questions to emphasize best practices for the ethical uses of AI and to promote the work of diverse research groups that span the perceived boundary separating AI and healthcare research. The program now includes 4 centers that will generate hypothesis-agnostic data sets for functional genomics, voice as a biomarker of disease, AI and machine learning in acute care, and human health restoration after disease. The program also includes the Bridge Center for Cross-Program Coordination, which is responsible for integration and dissemination of the activities, knowledge, and practices from all the other components of the program. And in April, the consortium held its first in-person meeting, in which I participated in a fireside chat along with the 4 other co-chairs of the program, including the directors of the National Institute of Biomedical Imaging and Bioengineering, the director of the National Library of Medicine, the director of the National Eye Institute, and the director of the National Center for Complementary and Integrative Health. The Somatic Mosaicism Across Human Tissues, or SMART network, is a new NIH Common Fund program aiming to build a greater understanding of somatic mosaicism, which reflects the unique post-conception changes in our DNA that can vary from cell to cell. Now, key aims of this network include developing a catalog of somatic genomic variants, creating an innovative DNA sequencing analysis tools, and forming a data workbench that integrates with other data sets. Now, last month, the NIH Common Fund announced the first set of awards for the SMART program in response to 5 funding opportunities. The Tissue Procurement Center was awarded to the National Disease Research Interchange, the Data Analysis Center was awarded to Harvard Medical School, and the Organizational Center was awarded to Washington University. There are 5 awards for Genome Characterization Centers, specifically at Baylor College of Medicine, the Broad Institute, New York Genome Center, Seattle's Children's Hospital, and Washington University. Furthermore, there are actually 12 institutions that received the Tool and Technology Development Awards that round out the first set of awards for this new Common Fund project. Now, the All of Us Research program is seeking to build a national resource cohort of over 1 million or more participants, reflecting the diversity of the United States. The program is creating a partnership with participants in order to advance precision medicine and to change healthcare for the benefit of all. Now, last month, we were delighted to see that the program expanded the data available in the All of Us Researcher Workbench, including the release of nearly 250,000 whole genome sequences with participant consent for broad research use. 45% of the data are from people who identify as members of a racial or ethnic group who have been historically underrepresented in biomedical research. Researchers can now access information from over 400,000 individuals, including data from survey responses, physical measurements, and electronic health records. The data releases also includes long-read genome sequences from over 1,000 individuals. We are obviously excited about this update, strongly encourage members of the genomics research community to visit the All of Us Researcher Workbench and to start exploring and analyzing these data. And access to the Researcher Workbench is available to researchers from eligible organizations after they complete appropriate training. The goal of the NIH Cloud Platform Interoperability, or NCPI, effort is to establish and implement guidelines and technical standards to create a trans-NIH cloud-based federated data ecosystem in order to facilitate end-user analyses across participating cloud platforms. Now, NCPI was launched in late 2019, and it done so in collaboration with our NHGRI, Analysis, Visualization, and Informatics Lab Space, that Anvil program I told you about earlier, but also other cloud-based programs and resources supported by other parts of NIH, including the National Cancer Institute, the National Heart, Lung, and Blood Institute, the NIH Common Fund, and also the National Library of Medicine. Now, this past January, the NIH Office of Data Science Strategy awarded funds to Research Triangle Institute to establish an administrative coordinating center for NCPI. This center will provide technical, administrative, and coordination support for the NCPI program. And then in March, the Administrative Coordinating Center released a solicitation for interoperability projects to test or implement tools and approaches with the aim of facilitating researcher-driven analyses of data sets across the NCPI platforms and repositories. And the applications for this solicitation are actually due today, May 15th. So with that, let me move on to my second to last category and discuss some things in the areas of communication and policy and education. And let's first start with a nice celebratory summary about National DNA Day. So as many of you are aware or were aware, April 25th is National DNA Day, and this April 25th was a particularly special one. So NHGRI actually hosted a symposium to celebrate two specific milestones. First, the 20th anniversary of the completion of the Human Genome Project, also coinciding with the 70th anniversary of the discovery of DNA's double helical structure. Well, organized by our Education and Community Involvement Branch and our Office of Communications, the event brought in close to 100 in-person attendees and just over 700 virtual attendees. I had the privilege of leading a panel discussion with several of my fellow NIH Institute and Center and program directors, exploring how genomic advances have directly influenced research at their respective institutes and centers, or in Josh Denny's case, the All of Us Research Program. But other highlights of the day included discussion about the benefits and challenges of using population descriptors in genetics and genomic research, a presentation highlighting an international celebration of DNA Day in Ghana, and a panel showcasing the diverse career opportunities in genomics. The day concluded with Francis Collins presenting the annual Louis M. Slaughter National DNA Day Lecture, and not surprisingly, his lecture concluded with Francis singing a song, which is all available to see on YouTube. So that wasn't my favorite part of the day, because I especially enjoyed a surprise segment in which I challenged my predecessor, that Collins guy, to a series of activities to determine who is the greatest of all time, or GOAT, NHGRI director. Now, that spirited competition, which you see a slice here, it actually consisted of a pre-recorded video, but then was followed by a live segment at the symposium itself. That's the photo you see there. Now, together, the complete GOAT competition takes about 17 minutes to watch on YouTube, where you can find it at our Genome TV channel. And that link is available, document 42. But I really want to entice you to watch this. And so to see some of the green Collins frivolity, I'll just show you a brief teaser.
Just wait, Green. Just wait, man. You're gonna be so sorry you took me on. Collins, he's so 90s. He's not the GOAT. He's so yesterday. He is so the behind of genomics, not the forefront of genomics. It's not even gonna be a competition. You're gonna lose this so bad.
Not a chance.
Green, I don't know. DNA Day for you is gonna be such a disaster.
When you're the GOAT, you're the GOAT, and I'm the GOAT.
Ha!
I'm the genome GOAT. I even have a G on my back, both for genome and for
yeah.
GOAT. Absolutely telling you that's what it's all about.
Save this for the competition, you guys.
Okay, I'll see you in the competition, Francis. It either goes downhill or uphill from there, depending upon your perspective. Now, it really is a 17-minute video on YouTube. And you council members, do not start watching it now and those remote. I know what you're thinking about, but I will give you a clue. If you don't go to document 43, that's the harder way to do it. All you have to do is go to Google and search under Francis and GOAT. Now, when I did that, the very first hit you get is a paid ad for a petting zoo where I think they have a goat named Francis. And it's a paid ad, so it shows up as number one. But number two, prominently featured, is the link to our 17-minute goat video. So you really want to, if you haven't seen it, you got to see it. And it was a lot of fun to film, as you might imagine. And actually, the outtakes are in some ways even better than what we show you, but that's a whole other story. What? Who won? I don't, no spoiler alerts. No spoiler alerts. You know, you got to watch 17 minutes of it to see who won. But I will just give you a clue. It was rigged, okay? Just, as far as I'm concerned, it was rigged. Okay. Now, speaking of frivolity, the NHGRI Office of Communications recently released two new Genome TV YouTube videos that are both fun and educational for everyone. Here in this first video, our social media specialist, who you may recognize as the referee for the GOAT competition, Jenny Montooth, and I show you step-by-step instructions for extracting DNA from strawberries using materials that are available in your kitchen. Now, this is actually an updated version of the original instructional video that I made for showing you how to purify strawberry DNA, and I made that with Carla Easter 12 years ago. A video, I may tell you, that has garnered nearly 500,000 views on YouTube. But it was time to update and give it a more modern flair, and so that's why we made a new one. In the second video, NHGRI researchers from Adam Filippi's group and also the NIH Intramural Sequencing Center show you how to extract human DNA, sequence it, and interpret the data, let's just say with some fun DNA hijinks along the way. So I would point you to those two videos. Now, in a video that's not released yet, but will be, is associated with a little slice of history in line with celebrating the 20th anniversary of the completion of the Human Genome Project. And as many of you know, the story of the 13-year Human Genome Project is a tale of great individual and group accomplishments and in many ways of incredible audacity. The project actually consisted of many smaller narratives, some scientific, some political, and some sociological. So to capture some of this history as part of the 20th anniversary celebration, NHGRI recently recorded a virtual discussion including the major leaders that pushed the Human Genome Project across the finish line in 2003. That group, and some of you remember, was dubbed the G5 and included the leaders of the 5 genome sequencing centers that produced the largest amount of sequence data. Well, for this incredibly fun virtual reunion, I was joined by Jane Rogers, then at the Wellcome Trust Sanger Institute, previously known as the Sanger Center, Michael Morgan, who was then a leader at the Wellcome Trust, Ari Petrinos, representing the efforts of the Department of Energy's Joint Genome Institute, Eric Lander of the Broad Institute, Whitehead Institutes for Biomedical Research, Richard Gibbs of Baylor College of Medicine's Human Genome Sequencing Center, Bob Waterston, of course, then at Washington University School of Medicine's Genome Sequencing Center, and finally Francis Collins, who was then NHGRI director. Now, while a full reunion video is not available yet, because we're actually enhancing it and putting in a lot of fun old digital archives we have, photos, videos, and the like. There actually is a teaser video that you can watch that's available already on YouTube. But we hope to get this out in the coming weeks, and I think it's going to be really a wonderful thing for you to watch to learn yet more history about the Human Genome Project. And yet also sort of tied into celebratory things, it actually relates more to 10 years ago when we celebrated the 10th anniversary, because some of you may recall that when we did celebrate the 10th anniversary of the Human Genome Project's completion, NHGRI and the Smithsonian National Museum of Natural History developed a collaboration to create Genome Unlocking Life's Code exhibition. And the exhibition examined the complexities of genomes, particularly the human genome. It chronicled the remarkable breakthroughs that have took place since the completion of the Human Genome Project in that first 10 years. And it highlighted both the benefits and challenges that genomics brings to modern society. And after a year-plus stay at the National Museum of Natural History, the exhibition traveled to 12 US states and also in Canada, and then returned to Washington, D.C. in October of 2021 for a final stay at the museum of its origin, and it attracted millions of visitors over the last 10 years. Well, upon closing of the exhibition, which actually finally closed in November of 2022, NHGRI was provided to come take custody of any of the pieces of the exhibition. And so the 5 pieces of Genome Unlocking Life's Code are now on display on NIH's Bethesda campus. Specifically, you can see 4 of them actually are in Building 31, where my office suite is and a lot of other directors' office suites are. And then one of them is also on display in the medical library in the NIH Clinical Center. Now, seemingly out of nowhere, to be honest with you, and perhaps related to the 20th anniversary of the completion of the Human Genome Projects, and I don't know if this happened to any of you, it absolutely happened to me, genomics has become a very hot topic for students participating in National History Day activities. So for the first time ever, and I thought it was wonderful, I was contacted not by one, but by 2 student groups seeking Zoom interviews with me and other genomics leaders as part of National History Day projects, specifically focused on the Human Genome Project, which I just thought was way cool. So here are 3 delightful and spirited high school students from Boulder, Colorado, who interviewed me by Zoom, and then they wanted more experts to talk to, and I ended up arranging for them to speak and Zoom interview Rick Myers and Richard Gibbs and Karen Miga. And then more locally, really within about the same week or so, I was contacted by 3 similarly delightful and spirited middle schoolers from Howard County, Maryland, who then interviewed me and Vince Bonham and Sarah Bates by Zoom, and there's Sarah and I standing with them in a photograph. Now, their extensive research allowed them to make this highly informative presentation on the Human Genome Project, and they proceeded to get the top honors at the state regional level, and then they competed at the Maryland state level just about 9 days ago, and that's the photo from 9 days ago where Sarah and I visited them at the state competition. And by the way, and you really may not have a very good view of it, but you should notice the ties that the gentlemen wore at the state competition, and they bought in bulk and gave me one, which I am proudly wearing today to commemorate their remarkable accomplishments in National History Day activities. So that explains the tie. And it also, I find it really heartwarming that it's really great that middle schoolers and high school students are starting to see Human Genome Project, Advancing Genomics as something part of the nation's history. And it's right up there with all the other obvious things we think about with history, but this is a really important part of our history. Speaking of history, what a great transition. The NHGRI History of Genomics program's digital archive is set to enter an exciting new phase in the coming months. The group has been working on some new tools and resources that will allow greater access to some of the unique historical assets in the Institute's archival collections. In anticipation of these developments, we just wanted to show you a few items from this archive, but I'll also tell you as a teaser, assuming the schedule doesn't change, we're actually going to have a special presentation about this at the September council meeting. So just to give you, again, a little bit of a teaser. This first item is Francis Collins's handwritten notes for the June 2000 press conference with Craig Venter announcing the completion of the draft human genome sequence. This is just one document from nearly 100 boxes of digitized materials from Francis's collection, which he accumulated during his tenure as NHGRI director. The three other items are actually documents from Elka Jordan, accumulated during her tenure as NHGRI Deputy Director from 1988 to 2002. The first is actually a draft sketch from 1988 of a possible budget and timeline for the Human Genome Initiative, or something that would soon then be renamed the Human Genome Project. Next is actually the seating chart from the very first meeting of the Program Advisory Committee on the Human Genome in January of 1989, just before the Human Genome Project started, maybe a year before. And last is the official meeting report from the February 1996 Bermuda meeting, in which the principles of data sharing and open access to genome sequencing data were laid out and adopted. So just 4 examples of hundreds and hundreds of thousands of documents and pages we have now electronically archived. So we'll talk more about this in September. Meeting in person for the first time since 2020, the Inter-Society Coordinating Committee for Practitioner Education and Genomics, or ISCC-PEG, held its 12th annual meeting in February. At the meeting, I gave a talk highlighting recent NHGRI achievements, but then after my talk, Josh Denny, the Chief Executive Officer of the NIH All of Us Research Program, provided an update about that program, including discussing the release of participants' genomics health-related results. Then presentations were given by the second class of ISCC PEG scholars describing their ongoing genomics education projects. ISCC PEG members continue to develop resources to integrate genomic literacy into clinical practice. 10 different project groups held active working sessions, including both in-person and virtual participation. These discussions included updates on the direct-to-consumer genetic testing resource for healthcare professionals, as well as the soon-to-be-released materials answering common questions that nurses may have about becoming a genomics nurse and integrating genomics into their practice. And videos of the presentations and slide decks from the meeting are available on the ISCC PEG website. And last update in this section, Beth Tuck was recently selected to be the new chief of the Education and Community Involvement Branch within NHGRI's Division of Genomics and Society. Beth is currently the Executive Director and Director of Science Education at GenSpace, the world's first community biology lab in Brooklyn, New York. As Executive Director, she's overseen the organization's staff, program strategy, fundraising, and finance, and she's also served as a key liaison and spokesperson for GenSpace. Beth will actually be coming home to NHGRI, since she was previously a genomics education specialist within the Education and Community Involvement Branch. Her earlier work at NHGRI focused on enhancing genomic literacy, education, and community engagement at the K-12 and college levels. And this work included designing curricular resources, engaging students and teachers in hands-on genomics activities, supporting teacher professional development, and training scientists in public engagement skills. Beth initially arrived at NHGRI as the very first participant in the ASHG NHGRI Genetics Education and Engagement Fellowship. So once again, this illustrates how some of these fellowships we have, they bring people into the genomics space and they go on to do various things. And sometimes they even come back to us to take leadership positions or other positions. So Beth will actually begin her new appointment at NHGRI in the early summer, and we are excited to welcome her back. And finally, let me just say a few things about our intramural program, but really saving most of those things for the presentation that will follow mine. One thing we're very proud of, Adam Filippi, Harang Rai, and Sergey Korin of NHGRI's intramural genome informatics section in the Computational and Statistical Genomics branch are finalists for the Samuel J. Hyman Service to America Medals, also known as the SAMMYs. Now, the SAMMYs honor the remarkable and inspiring accomplishments of federal employees. And this group was nominated for their work in, guess it, seeing what their thing is, T2T, of course, the telomere to telomere consortium, which successfully generated the first complete assembly of a human genome sequence. So congratulations to Adam, Harang, and Sergey. And we are rooting for them to win it. We'll see what happens with how the voting comes in. And to set up our next presentation in some ways, I did want to point out that NHGRI Scientific Director Charles Rotimi was featured on the cover of the March issue of the American Journal of Human Genetics. And this eye-catching cover pointed to the issue's publication of Charles's 2022 American Society of Human Genetics presidential address, which was entitled One Human Race, Billions of Genomes. The address emphasized that as we learn to read and interpret information coded in the living and evolving genomic history book of billions of individuals from around the world, we are gaining insights into fundamental questions about our evolutionary history, health, and complex issues surrounding individual and group identity, including the notion of human racial groups. All really relevant for things we're going to be discussing later today. And lastly, I just wanted to tell you that once again, our intramural research program more broadly has been highly productive since the last council meeting. And just let me give you a few highlights that I know Charles won't touch on. Very recently, Diana Bianchi and her group studied an enhanced mouse model of Down syndrome that more precisely captures the features of the condition. The researchers found that the new model shows milder cognitive traits. These results have important implications for human clinical trials that aim to improve cognition in these patients. Elaine Ostrander and her group led a study that characterized the genetic ancestry of free-roaming dogs that are currently living in and around the Chernobyl nuclear power plant. The researchers sampled over 300 dogs and found large, complex dog families living in the area. This study establishes the importance of genetic studies in understanding the effects of exposure to long-term low-dose ionizing radiation. And Sergey, who you just saw in the previous slide as well, along with other researchers at NHGRI, designed and tested a software system called Verco, and that automates analysis methods used by the Telomere to Telomere Consortium to assemble gapless genome sequences. This software can assemble whole genome sequences in a matter of days and promises to lead to more gapless genome sequences from humans and other species. And with that, let me just remind you that we work hard to make all of our information as available to you as possible. And one of the ways we do that is through this very user-friendly, staying connected kind of page that on genome.gov. And if you just visit the URL shown here, I will greet you, but you should scroll down and immediately see these 9 major resources associated with the Institute. This includes links for genome.gov websites, also to my Genomics Landscape monthly newsletter, my Twitter feed, but also convenient links to our strategic vision, our brochure, our talking glossary, our Building a Genomics Workforce Action Agenda, our Genome TV channel, which I know you're going to dive into to see some of the other assets we've put there recently, and also our History of Genomics program set of oral histories. And if you want yet more, we also scroll further down, you'll see things of recent talks, recent podcasts of mine, and op-eds. So I hope you find this convenient if and when you ever need to find things on genome.gov. So let me thank all the many NHGRI staff members who contributed to the slides and associated materials that I just reviewed. Without such a group effort, I would look really bad, actually. And this is incredibly important to have it well organized coming into a council meeting. An additional thanks goes out, of course, to our communications group and for all of their help in making the electronic resource so organized for my director's report and for all the entire open session. And then a special thanks to the usual ringleader, Chris Wetterstrand, who helps me get this across the finish line. You have to search for a while to find her here. This is a historic photo of the International Human Genome Sequencing Consortium. This throwback photo was actually taken at the May 2002 meeting at Cold Spring Harbor Laboratory, and there's Chris Wetterstrand. You had a strain of Sierra, but she's in there, and thanks to Chris. And with that, I will draw my director's report to an end, and happy to take any questions you may have. Thank you. Any questions? I'm also looking remotely if anybody wants to raise their hand. Okay, move on, right?